We're very excited today to have with us Dr. Sonja Schrepfer and Dr. Cetrulo. They are coming together to present hypoimmune cells and xenotransplantation. And I will be dropping their recent paper into the chat as well. We will take some questions in the chat and we'll open that up at the very end of the talk. A little bit about them. Dr. Schrepfer is the scientific founder of Sinai Biotechnology and appointed professor at Cedars Sinai Medical Center. And Dr. Cetrulo, he is a vice chair of research department of surgery and regenerative medicine, also at the Institute of Cedars Sinai. Welcome, both of you. Thank you so much for joining us, and we really appreciate you taking this time, and we're really excited to hear what you have to say.
Thank you for having us.
Thank you, Monica. Thank you for having us. And yeah, I share the slides.
Yes. I can see, oh yes, perfect.
Awesome. Great. Okay. Yeah, we are happy to discuss today hypoimmune cells in xenotransplantation. And Kurt will show you the promise, and I more will show you the challenges with genetic engineering, etc. So the goal is really what we did in the past and still doing is how can transplant cells, tissues, organs, usually from one person to another without the need of immunosuppression. And we are using what we call the hypoimmune cells. What that really means is that we use genetic engineering, we modify the cells, and then we transplant them with the goal that they evade the immune recognition, meaning that the host immune system doesn't see that we transplanted the cells. But in the future, of course, we want to go more complex tissues, more organ transplantation, and that is really, really promising with the field of xenotransplantation. So that's why we really would love to not only now focus on allogenic transplantation, but really tackle that xenotransplant field. And you will see from Kurt where that field is. I personally couldn't be more excited about that. So I thought I start as a background a little bit what that hypoimmune engineering means, and then I will show you the challenges, how that might be able to translate into xenotransplantation, but with tweaks. So that system is not there yet. So what we need with hypoimmune, and I put here from one of the publications, a modified figure where we really worked with islet cells. We are really interested in the type 1 diabetes space. So it was an islet cell. And when you transplant that from a donor to a recipient, you get immune rejection, and we knocked out the beta 2M and C2TA. So we removed the MHC molecules on the cell surface of those cells. And that we do because they act as the fingerprint. So when you remove the fingerprint, the adaptive immune cells, the T cells, they don't see that you transplanted a cell that has a different fingerprint. What I also like about knocking it out and not just knocking it down is that those MHC molecules, they are presenting neoantigens, MIHA, so minor histocompatibility antigens. And when you knock them out, you don't have that presentation to the immune system at all. But because then the innate immune cells, macrophages and NK cells, they are in our body to sense what we call a missing self. So that is a cell without fingerprint. They would attack immediately. And what we did in the past is that we overexpressed a molecule that was called or is called CD47. And that can inhibit innate immune cells that they don't kill when they are triggered through a missing self. So that work has been published. We will not go into those published data because we want to focus on xenotransplantation, but I thought it makes sense to highlight the last publication, and that was with a PI, Professor Carlson. He is the principal investigator at the Uppsala University. And what was done is hypoimmune engineered islet cells. So they didn't have the MHC molecules. They did have the CD47 overexpression. And they were transplanted into the forearm of a 42-year-old patient. You see that here, the transplantation itself. And the patient was type 1 diabetic since his childhood. And he had no measurable C-peptide. That was an inclusion criteria. And what you see on the graph on the lower here, on the lower row, is that before the transplantation, there was no measurable C-peptide. And then the x-axis shows you the days after the transplantation. The publication reports up to 3 months, but you might have seen also now the 6 months data presented by Professor Carlson, and you see that there is measurable C-peptide. C-peptide levels are really low, but that was on purpose in that study because the dose was so low. Patient didn't have any response to a mixed meal. So you see at baseline here, C-peptide levels wouldn't go up, but he responded after the hypoimmune transplantation. So that is a nice proof of concept that you can use genetic engineering, modify cells, transplant them without any immunosuppression. The patient didn't get any immunosuppression, nothing that would alter the immune system, no steroids, etc. Was pure injection of the hypoimmune islet cells. So in the picture here, you see his arm at 3 months, and that was a PET MRI. So you see that the islets in the arm are lighting up in that image. And why I show that is because this is so promising that you would think, great, that can be now translated into all the other fields, like going from allotransplantation to xenotransplantation. And unfortunately, that is not as easy because now you switch your donor to a pig donor, and the pig is really attractive because of the organ sizes, etc. We can produce engineered pigs, etc. So the pig is very attractive. However, there comes a whole new hurdle in xenotransplantation. And the question now is really, how can you use hypoimmune editing to make a pig transplant work? Because then now you can think not only about cells, you can go into tissues and even organs, and that is of course very attractive. But before I go into the challenges of that hypoimmune editing, I hand it over to Kurt to really show you the promise of the xenotransplant field.
Thank you so much, Sonja. Xenotransplantation has always been quoted as the future. It is the future of medicine, the future of transplantation, and always will be. But that statement, I think, is now crumbling as we've seen. So I had the privilege of working in David Sachs' center at Mass General for many years with this group. On the right there, you see Tatsuo Kawai, still a collaborator of mine, and they have worked on xenotransplantation with gal knockout swine. As you remember, the wild type swine has a sugar on all its cells that we are reactive to in the human body because of bacteria in our body. We make preformed antibodies. So a normal pig heart or kidney, when you put it in a human, hyperacutely rejects. So that was sort of the start of genetic engineering in xenotransplantation. Other groups have gotten involved, such as United Therapeutics and eGenesis, and they've had long-standing collaborations with these institutions with many knockouts of these non-carbohydrate antigens on the surface of the cells. But long story short, the first successful swine kidney that was transplanted in March 2024, and a second case done recently at Mass General, and 2 other great cases done at NYU. The patient at NYU actually did very well for up to 4 months before requiring dialysis and explantation for a number of reasons of really rejection. But again, it's a proof of concept. Really, really exciting work. Heart and lung transplants have also been done recently at NYU in Maryland, and a lung was recently done in China, which is the patient's doing quite well by all reports. So the resources that have been put into this field now are really finally coming to fruition. There was a big pause in xenotransplantation for many years with what I consider unfounded concerns about retroviruses that turned out to really not be very important or meaningful. So now we're off to the races. Clinical trials are now funded and ongoing at MGH and NYU, and we're looking forward to this new age, golden age of xenotransplantation. However, there are still problems to solve with this. Can you advance the slide for me? Thank you so much. So before we talk about that, I just want to tell you, so when I joined David Sachs' lab, he had gal knockout swine and was using the skin from these swine to help soldiers. That was his idea, to put them on to burn patients and have them be temporary skin coverage, just as we use aloe skin for coverage. Well, now we routinely use this product about a decade later in wounds such as this. This is a sweet, healthy, otherwise healthy little old lady who lost the skin on her leg from an infection, and they wanted to amputate this leg, which is basically, the mortality of when you had an amputation at 86 years old, you stay in bed, you get diabetes and bed sores, and your mortality is about 60% over 2 years. So we used a gal knockout swine skin. If you want, I can advance here. In the OR, this is a approved product by the FDA now, where we're able to take these with no donor sites now and place it on her leg. Next slide. In the OR, and you can see this incredible. They come in quite small pieces at this point, but they heal quite well. And this is just creating a wound bed. I won't say that this stays on and leads to this result. We do have to go back and autograft with the patient's own skin over this, but it covers tendons, bones, exposed nerves, things that you couldn't cover with just a skin graft. And we're able to save this woman's leg. And it was really a remarkable clinical result. And if you look at the next slide, we've done this now in a few patients, and you can see the results that, even secondary healing is better than a normal allograft with this technology. So exciting proof of concept, and not quite a kidney or an organ or heart or lungs, but our humble plastic surgery contributions are still important and still can matter to patients. So next slide. So we are, again, this just summarizes really the basic knockouts that have been used, and many other knockouts in these swine have happened. But still, there's a real level of rejection. The xeno barrier is quite high, and I think we're not going to really progress to lower immunosuppression levels or even tolerance until we get the kind of edits that Dr. Schrepfer is going to describe. So I think it's a critical junction in clinical xenotransplantation right now. The clinical trials are happening. We've seen that we can put these organs in patients, and they can do quite well. And I think we need to take the next step with these gene edits. So with that, I will turn it back to Dr. Schrepfer to talk about how she plans to solve this huge barrier in xenotransplantation, but not so huge anymore that we have these edits.
Thank you, Kurt. Yes. Yeah, and we are excited to come back and report on our progress. What I will show you now is where we are as of today, how we are thinking about to tackle it, and we also wanted to show you the challenges. So we had to learn a couple of things, of course, when you move from the allotransplant field to the xenotransplant field. And we did that based on the hypoimmune edits I showed you. That's why I thought it's valuable to show you the background. And we are working here, when I write EC, with endothelial cells, not with islet cells, although we will now transfer to islet cells. But the reason for that is endothelial cells are highly immunogenic. They're antigen-presenting cells. So if you have a high bar cell type to study with, it's easier to translate it later to other cell types. So that's why you will see in that publication that was shared and on the slides, the endothelial cells. We love them for a first proof of concept as a high bar study. So the first thing we did is, of course, we removed the MHC or HLA molecules for the reasons I explained already, to overcome the adaptive immunity. And usually what happens, and that was published now so many times, including in the Uppsala trial, that the human innate immune cells, they are drifting away, they're not attacking. They are activated with the missing self when they see there is no HLA, but they're inhibited and they will not attack a cell that overexpresses human CD47. However, in xenotransplantation, and we chose here for the first experiments the model human cell into monkeys, into macaque monkeys, you do see here that an immune cell from a macaque monkey doesn't react to human CD47 overexpression. So it's simply a learning. We had to learn the hard way that there is no cross-species recognition. So you cannot overexpress the human CD47 and then expect that the macaque immune cell would be inhibited. So the next step was, let's see if we can overexpress the macaque CD47 on a human cell. And what really turned out to be true is that, yes, if you stay in your species and you express the CD47 of your host species, then the system really works nicely. So we do have now a tool where we know, as long as we stay with the species of the host, that we can use that molecule also for xenotransplantation. And you do see here the data. So we had the human endothelial cells, and when I write here HIP with that star, what that means is they have the hypoimmune edits, so the MHC class 1, class 2 knockout, and then the star means the expression of the host CD47. So in that case, it was a macaque CD47 overexpressed, and then they were transplanted into a macaque monkey, so xeno, crossing the xeno barrier. And what we learned is when we did the blood draw after the transplantation in the monkeys, for the T cells here, that the T cells were not activated because we had knocked out the MHC molecules of the transplanted cell. And how you read those graphs is, so you see here the percentage drop of the cell index at 90 hours. And the cell index itself shouldn't drop. So we are doing basically over 90 hours a killing assay on our platform where we can measure if the cells are surviving or if they get killed by the effector cells. In that case, we added the T cells. And what you see is in gray, before the transplantation, nothing happens. The immune system hasn't seen the transplanted cells. But 7 days later, you see here in blue, also the macaque T cells are not recognizing the transplanted cells, and that is because of the MHC knockout. So you want to have that here at 0. And then you see for the macaques NK cells as well, when we overexpress the macaque CD47, there is no activation. The NK cells are not killing the missing cells. Same for the macrophages, and same was true when we run an ADCC with NK cells or macrophages. So we really were able to address that innate immune killing in a xenotransplant model by just using the host CD47. However, we run into severe issues with another innate immune cell population, and that were the PMNs. So polymorphonuclear cells are usually not recognized in publications. If you look at detailed immune analysis, you will hardly find any team, any group that studied the PMNs. And that is okay, most likely for the allogenic field. However, in xenotransplantation, we learned that it is a major issue because what the PMNs are doing, they get activated mostly through the toll-like receptors, the 4, 7, 8, 9, and they get activated by DAMP signaling, by xenoantigens, and by cytokines. So this is a huge issue because those cells, they kill immediately. They are like an innate immune killing. They don't even have to learn. They get activated, they kill. And what you see here is that even before the transplantation, the immune system doesn't need to learn about the transplanted cells. They're killing immediately. And they did so also at the day 7 after the transplantation. So we had to find a way to tackle the PMNs, which is one additional layer of complexity if you want to go into hypoimmune for xenotransplantation. So what we did in the beginning was, first question was, can we inhibit those PMNs in any way? So we thought about a cocktail of drugs that have been used in the clinic. They were known to have some effect on PMNs, and we used them in quite high doses. So we transplanted the human hypoimmune endothelial cells into the monkey, but because the PMNs had killed the cells, we inhibited the PMNs with those 5 compounds. And you do see here, if you're interested in the 5-drug regimen, you see in the publication in more detail, and I named them here. But what they are mainly doing is they can inhibit the toll-like receptor pathways and kind of inhibit the PMNs. But what we learned is when we transplanted on the top row human wild-type endothelial cells, so no editing, just the human endothelial cells into the monkey, and we gave those drugs in a single dose, so monotherapy, you see the cells are all killing our transplant, so 100% killing. When we mix the drug and we had a 5-drug regimen, then you do see that it works, the PMNs seem to be inhibited. However, because the transplanted cells were not genetically modified for hypoimmune, the PBMCs of the monkey now killed the cells. And that is expected. That is basically the allogenic, the adaptive immunity coming in, plus then the innate immunity in addition. So you do see that the 5-drug regimen is not affecting PBMCs. And you do see that we need still the hypoimmune edits, because on the bottom row, when we have now the hypoimmune endothelial cells transplanted, usually the 5-drug regimen inhibits the PMNs. And because we did the genetic engineering, the PBMCs. So the PBMCs contain the T cells, the NK cells, and a bunch of other cell types, but you see that the rejection process here is inhibited by the genetic engineering. If you look into the publication, we couldn't run those studies long term because that 5-drug regimen we are using here is highly toxic. So this is not a path we can offer to our patients. It was really just to understand, can we even inhibit PMNs? Is that the way forward? And if you look here at the bottom row, we think that with hypoimmune engineering, controlling the PBMCs, plus additional edits to control PMNs, we are a step further in really making xenotransplantation a reality. So we knew now that drugs we will not move forward. So how can we translate that and use that knowledge and then transfer to genetic engineering? So we now switched the model because later we will not transplant humans into monkeys, we will use the pigs into humans. So we switch here the model where we are now going to pick endothelial cells, and we studied that with human immune cells. And what you see here in the pig endothelial cell is that instead of the MHC, you have to knock out the SLA. It's a swine leukocyte antigen, but similar to the human the HLA, you can easily knock it out. And then we had to overexpress the human CD47, so from the host. And we could show that the human innate immune cells are indeed now inhibited. And I show you that on the next slides, but we had to address the PMN issue. And we did that by overexpressing of CD200 and 99. Those are inhibitory signals. And they could, that was the hypothesis, maybe inhibit the PMNs, that if you get the activation of the PMNs, you have that overriding inhibitory signal. So that was the goal of what we wanted to achieve, to get a step closer to clinical reality. What we did here is we had 3 different pig endothelial cell lines. One was wild type, so normal pig endothelial cells, no editing. The other one was the endothelial cells from the pig, and we only overexpressed CD99 200, so the 2 molecules we identified to be important for the PMNs. And then we transplanted or investigated the pig hip with a star. So what that means is SLA knockout, then the human CD47 overexpression, and the CD99 CD200 overexpression. So you have now a couple of more edits in those cells, but let's see how the immune system reacts. So the human immune system, when you incubate that with the pig wild-type endothelial cells, you see that the PMNs here, they are killing as expected. We have seen that before, and the PBMCs are killing. So that is really the fulminant xenogenic rejection you are seeing here as expected because the pig endothelial cells didn't have any modifications. When we then did the overexpression of CD99 200, you see that we can indeed control the PMNs. In none of the conditions we did was the PMN killing anymore. So CD99 and 200 are really nice inhibitory signals for the PMNs. But because we didn't have the other hypoimmune edits, you see that the human PBMCs were still rejecting those endothelial cells. So then we went a step further and did the hypoimmune edits I showed you. So SLA knockout, CD47 overexpression of the host, so human, plus the CD99 200. And now you see that the PMNs are not killing and the human PBMCs are not killing. So we do think in those areas that we can use the hypoimmune editing and the engineering strategy to really overcome that xenogenic barrier and rejection. As Kurt already said, that's of course not the only story. You still need the alpha-gal, etc. You need a couple of more molecules, but everyone studied the alpha-gal, and we think where we were trying to find where are the holes, where do we still need work to really then modify the cell that it can overcome that really high bar rejection barrier in xenotransplantation. And we do think that the future of xenotransplantation is really using genetic engineering and having really hypoimmune cells with the goal of limit the immunosuppression really low dose, or even one day. That would be my personal dream, to go immunosuppression free. And what I like about that field is that you can think about whole organs. You're not limited to only cell transplantation. So, and Kurt showed it already very nicely with skin grafts, that you can really go for more complex structures. And the xenotransplantation field looks like, as Kurt said, it's not moving a lot. I started in med school when I was in Munich under Professor Bruno Reichert in xenotransplantation, and I observed that field for a long time. And I do think there are small steps, but they're really persistent. And I think if we keep that persistency up and we have enough teams tackling that issue, that xenotransplantation will have a future, and genetic engineering and hypoimmune I'm excited to see where the way goes for not only allo, but also xenotransplantation. Thank you.
Thank you very much to both of you. It was a fascinating talk. And we do have a few questions already in the chat, and I'll just go ahead and read them out for those that listen to this instead of watch later. From Klaus Petkold, are you using hip organs or organoids, in fact, creating an immunoprivileged site or niche that may in itself present problems longer term?
Yeah, so the goal is, of course, to create not a niche, but to have an organ hypoimmune or cells or tissues. And of course, we think about safety a lot, and there are a couple of safety issues you have to think through. Will it become a viral reservoir is always one question. But we do have a paper in iScience where a whole mouse was created that was hypoimmune. That mouse didn't seem to have any issues in any regards of health concerns, tumor development, more viral infection. It was housed into normal housing. However, yes, you would need to think about a safety strategy or really have a robust package to understand that. With cells, when you do the genetic engineering, that always has some risk with it. It's really a risk-benefit question. But for the xenotransplant field, I think we can think about organs, etc., that will be safe. And there are the concerns, and more work needs to be done, no question. But we also know now that a better 2M knockout cell, etc., can still clear viruses. There are enough publications now out in that field.
Yeah, I would just also mention the concerns about porcine endoretrovirus or PERV have been made a big deal of for a very long time. We actually there are many background swine that has PERV knockouts, but even whether that's necessary is even in question. We have an FDA approval for use the skin that we use. For example, we had safety concerns. We did many assays at many times and found no PERV transmission between the swine skin and the host. And if you go back to the original work on that, it's very difficult, even in vitro, for this virus to infect human cells. So it was something that's I believe it's been accepted as a real risk-benefit ratio, as Dr. Schrepfer said. So I think there's certainly other infectious diseases in any transplantation we should be aware of, and safety assays are obviously critical. But I think the benefit of the organ being able to be transplanted is far outweigh these risks that have turned out to be minimal to none. It's actually quite remarkable how little downside there is as far as infectious transmission of these porcine concerns so far.
Great. Here's another question from Dhanashri Survey. How long did the transplant cells stay? What is your perception on the risk of tumor formation using the genetically edited hypoimmune cells?
Mm-hmm. So to the first question, the study I showed you with the PMN inhibitors, the 5-drug regimen, was really hard to do in the monkeys. It is so toxic. So if you look in the publication, we pulled it after 2 weeks. So we had certain times in the paper, but nothing of that can be done long term. So we're planning now with Professor Cetrulo together studies where we're now really looking on pig cells with human and do the transplants there. So in the model I showed you, human to macaque, was really hard. So that's why in the paper you only see the short-term data. That being said, those innate cells and the PMNs in that case that gave us the trouble, of course, they act immediately. You don't even need any education, no adaptive memory. So they would kill immediately. That's why we feel 1 day in, you would already see the killing. That wouldn't change over time, but for the upcoming experiments, we will change the model and we will not go with the drug regimen. We will do everything with genetic engineering. For the tumor formation, the molecules themselves are not tumorigenic. It's really only when you do the editing. And of course, when you generate the pics, you will have a strategy to make that the safest way. What we did, the paper I showed you and the data I showed you, we do it really fast. So we don't look for safety. So meaning we use LVV, just put it in. We don't do a safety package because we need proof of concept at the moment. But down the road, those cells, the only risk is that with the engineering itself, that you hit like off targets or you hit something that will increase the risk for tumor formation. The molecules themselves, if you overexpress them, will not do that.
Great. Question from Jane Buckner, wondering if excuse me, autologous APC later present alloantigens as cells from this transplanted organ die. Is that a longer term problem? Is the question.
Go ahead, sorry.
No, let me repeat the question that I got it right. So autologous, so if you do autologous cell transplant, if there can still be rejection, that's the question, yes. And we did show that already. If you have an autologous transplantation and you induce, for example, mutations or some SNPs, and that is especially true when you do like iPSCs and like the Chinese colleagues are doing, and then you do iPSC autologous transplantations with islets, for example, you have a high risk that you introduce those mutations, and those mutations will be shown to the immune system because the MHCs are intact in those transplants. And that's what I mentioned earlier. I like that we are knocking it completely out. If you have only a knockdown or you don't modify it at all, you get the presentation of those neoantigens. We have 2 publications in that field I'm happy to share, Monica, with you, where we show that those called neoantigens will get recognized by the immune system. So it can really give you a rejection issue, although it is autologous. Yes.
Great, good answer. Any further questions, or would anyone like to unmute themselves and ask a question directly? I'm just checking to see if anyone has their hand up. Yes, Jane, please.
Yeah, hi, just thank you, Sonja, for that interesting talk. I guess my question was not about autologous transplant. It was about using your transplant with allo engineered, as you have, which looks beautiful. But once you have that transplant established, if you then can have induction of an inflammatory response around the organ, because you can present, you know, the neoantigens from that organ or the alloantigens through autologous APCs that are in the site to autologous cells. So it wouldn't be a targeted attack on the organ, which you've protected from. It would be more an inflammatory response in the region because of exposure to those antigens.
Yeah, that's a great question. And I do think, I mean, in the Uppsala trial we showed you, there was in the beginning for sure an inflammatory response. After the surgery you do, there was no steroids given. There were some wild type cells, so non-modified islets in the graft. I personally liked it because it will enhance your neovascularization. So I think it's good. It is not targeted, as you said, against the hypoimmune cells. So as long as you have the inflammation and it resolves after the initial surgical trauma and the wild types were removed, the hypoimmune cells can survive. So we thought a lot about it because the afferent presentation can occur through the APCs, for example, of the host, but the efferent way is really blocked. So although you have maybe locally some inflammation in the beginning, it didn't harm the graft, and it shouldn't. And if that occurs long term, I don't have any data on that, because the patient is 6 months in Uppsala. So I'm not talking data-based, but just my personal opinion is that even down the road, if there would be an inflammation, as long as the hypoimmune edits are stable and they don't show themselves, so the HLA wouldn't come back, it should not be any problem. But that is said without any data because we don't have those long-term studies.
Yeah, just a comment. I think it's a great question. You know, the local inflammation from a very small, pretty, you know, benign injection of islets is probably different than a kidney transplant, you know, as far as inflammation operatively. But, you know, we do a lot perioperatively to try to keep that inflammation down as much as possible. And there's a whole avenue of research looking at the suppression of the innate response that then triggers the adaptive response around a transplant. All those things will have to be considered. Induction regimens, if necessary, would be directed at just exactly that. So I'm sure there's certainly more to be done, but it already goes to show the kidneys that have been transplanted into humans that are swine have already done pretty well through their standard induction regimens, and got through that perioperative 30 days of inflammation and looked pretty good for a few months at least, until other rejection mechanisms take over. So I think that clinical hurdle is definitely achievable in clinical transplantation, even presently.
Thanks for that ad. Another question from Klaus Peckold. Jane's question also possibly applies to B-cell sensitization. Do you see or expect antibody-mediated chronic, in parentheses, autoimmune diseases?
Great question. So in the Uppsala trial, you find it in the results section because there were wild-type cells mixed in, so there was a donor-specific antibody response. It was through the donor HLA, so nothing was binding to the hypoimmune. But your autoimmune question is very valid. I mean, the autoantibodies, like GAT65 or so, they see the peptide through the HLA presentation. So if you have the HLA knockout, they wouldn't see it either. So really, they are flying blind. But it is a great question, and chronic rejection, etc., is something we always have in the head, in the back of our mind, because it is something we fear in solid organ transplantation, and we don't have any long-term data from the patient. So it is something we need to monitor and tackle if it occurs. Yes.
But we've a lot of centers have added rituximab to the induction regimen as well, both in soft tissue transplantation and perhaps Xeno is even more needed for that, to prevent that priming. It seems to be very helpful, at least in the hand and face transplant literature. They're very low numbers, obviously, but it's an exciting finding. The patients who've had a rituximab induction have gone to really not shown DSAs at all, at least in a short follow-up of a year or so, both 2 in a row there. So that's an NYU experience.
That's great. Well, I think this is really some great news for this space, and we really look forward to your working together and more advances in this area coming from Cedars-Sinai. So thank you both very much, and we look forward to our next conversation.
Thank you for having us.
Thanks again. Have a great rest of your day. Bye-bye.